If you've ever experienced any of the following symptoms: fever, cough, shortness of breath, fatigue, muscle or body aches, headache, loss of taste or smell, sore throat, congestion or runny nose, nausea or vomiting, diarrhea. Well, if you have, then you've probably been infected with COVID-19. Now you may be wondering what you can do to protect yourself. There are options. You can completely avoid people and become a hermit. You can strap up and mask up, or you can get pricked and injected with a mysterious substance that somehow helps you from getting sick. This last method is commonly known as a vaccine and has been around since 1796 with the creation of the smallpox vaccine. But what are vaccines and how do they work? Vaccines can be broken down into 4 different subgroups. The general gist of the vaccine is to inject a person with an agent that elicits an immune response, so that when the person encounters COVID-19 in the real world, their body has already seen the infectious agent and has antibodies to fight off the pathogen. 4 types of vaccines are vaccines made from a whole virus, a protein subunit, nucleic-based, or viral vector. Whole virus vaccines can either be from live attenuated versions of the virus or an inactive dead virus. An example of a common live attenuated vaccine is the chickenpox vaccine, and an example of an inactivated vaccine is polio. Vaccines can also be made from a protein subunit that contain purified pieces of the pathogen that are known to elicit that pesky immune response. Hepatitis B has a protein subunit vaccine. Viral vector vaccines use a non-pathogenic virus as a vector for DNA that encodes information for a specified immune responsive agent, such as an antigen or a protein. It essentially hijacks the virus's method of injecting genetic material into human cells in order to produce a controlled immune response. Finally, the newest type of vaccine involves nucleic acids, or the building blocks of DNA and/or RNA. For some background information, DNA must be transcribed to a slightly different form of genetic information called mRNA that is used to code proteins. mRNA is the newest type of vaccine because there were initially logistical problems of getting the mRNA into the cells without being degraded. Modifications of the delivery system and stabilization of the mRNA with some processing allowed our body to translate the mRNA into a spike protein known to elicit immune response in some COVID-19 vaccines. The Pfizer and Moderna vaccines are the first vaccines to be widely distributed in the US and are the first FDA-approved mRNA vaccines. There is a plethora of vaccines of different flavors to choose from. Whole virus, CoronaVac, VIBP CoV, Covaxin, WIBP CoV. Protein subunit, EpiVac Corona, Abdala. Nucleic, Pfizer and Moderna. Viral vector, Johnson & Johnson, AstraZeneca, Sputnik 5, Sputnik Light, and Convidica. Below further highlights the differences between the vaccines in terms of number of doses, storage required, and efficacy rate. In addition to the vaccines listed above, other COVID-19 vaccines are currently in testing. The figure illustrates some new COVID-19 vaccines that are currently FDA approved and the categories in which they fall under according to the type of vaccine. So now that it has been clarified that not all vaccines are created equal, exploring the different tests a vaccine has to go through in order to be approved for public use. Now, how in the world were we able to develop and integrate a vaccine to the public in such a short amount of time when it usually takes years to get one approved? First, let's talk about the process. According to the CDC website, the general stages of the development cycle of a vaccine are exploratory stage, preclinical stage, clinical development, regulatory review and approval, manufacturing, quality control. All vaccines must go through a rigorous testing and an inspection to ensure it's safe. COVID-19 vaccines followed a 3-phase protocol as several stages were required before FDA authorization. Phase 1 of this process consists in the application of the vaccine to a small number of healthy adults. Each dose is managed carefully and the volunteers are monitored for any side effects and any type of immune response. During Phase 2, some conclusions are drawn from the studies in Phase 1, and if not safety concerns present, the vaccine is given to hundreds of adults who come from different backgrounds and have varied health issues. The results from the stage should provide more in-depth information regarding the short-term side effects and risks. These look at the relationship between the dose given and the immune response, and may issue initial insights into how effective the vaccines are. Ultimately, for Phase 3, the investigations are expanded to integrate thousands of adults from very distinctive ages and backgrounds, and administer them with the vaccine to establish a comparison between how many of those who received it were protected from the disease in contrast to those who received the placebo. The rapid spread of COVID-19 made the development of these vaccines an international priority. Researchers and medical experts had already made progress developing vaccines for other types of coronaviruses. They applied lessons learned after the 2003 SARS epidemic and the 2012 MERS outbreak. All this previously acquired knowledge was applied while ensuring the safety and effectiveness of the vaccines to meet the FDA's rigorous standards for manufacturing quality, efficacy, and security in order to create the vaccines we currently have available. At this point, you may also be wondering if in the process of elaboration and experimentation of COVID-19 vaccines, some of them did not reach the final success. The answer is yes. Many of the initial prospects developed were not permitted to continue or did not provide the expected outcomes. For example, CureVac is a German biopharmaceutical company that opened the world up to the potential of mRNA to treat diseases and create vaccines. Many scientists and investors alike had expected CureVac's candidate, which uses essentially the same novel mRNA technology as vaccines from the Pfizer-BioNTech collaboration and Moderna, which had more than 90% efficacy in their trials. However, CureVac only produced a 47% efficacy rate according to the Science.org. The FDA had targeted at least 50% efficacy, which CureVac has fallen short of in the interim analysis. The World Health Organization had said it was looking for at least 70% efficacy. Another example is Merck, a $190 billion company with one of the largest vaccine businesses in the world. They were working on 2 different vaccines. The first, which was unique because they used viruses that could replicate once they were in the body, would be long-lasting, one-dose vaccines. The other vaccine used measles virus, a type of vaccine Merck has manufactured for decades. Both vaccines, however, produce lower levels of antibodies against SARS-CoV, including binding antibodies and neutralizing antibodies, compared to those seen in the blood of individuals who have recovered from COVID-19. As you can see, the standard for a vaccine to actually get to the public is extremely high. Now that you know all the facts, how do you feel about the vaccine?